Good evening, everyone. Thank you for coming. This is the fourth event of the Fall 2017 Provost Colloquium Series, and I would, I'd like to welcome you, and I would like to first thank the person without whose support and active participation the series would not be possible, Provost Joe Bruno. Thank you very much, Provost Bruno.
My cue. All right. Well, first of all, thank you for being so flexible and moving with the venue. At a last minute, we had to change, but this is a wonderful room, and it really is apropos to the topic today. So I am thrilled that, first of all, you were all good enough to work with us as we changed it from, I think, beers to here. What happened is, just to briefly tell you, there was a slight conflict with, I think, a short play production with Stephanie French coming right on the tail of this lecture, and we didn't want to curtail any discussion, because the best part of the Provost Colloquium is getting the audience to participate with questions and answers. Now, I just want to briefly thank people, and that's partly what my job is to do as a provost. And I'm not going to ask anybody in the audience what a provost is, because not many people know. Or is there anyone in the audience that happens to know what a provost is, besides this person standing in front of you? Well, just so you know, it's also synonymous. I'm glad the associate provost knows who the provost is. That's Jeff Weber. The provost is another name in academia for the chief academic officer. So, or what I like to say, the buck stops here. So when all the academic programs, the academic colleges, Dale, the library, D2L, wonderful Luis Vidal of instructional design, it all winds up in my lap, particularly when things go wrong. And luckily, when things go right, I thank all the people, particularly the faculty, for the good work they have done. And thank God I'm mostly doing that, not the former. But the Provost Colloquium, or the Chief Academic Officer's Colloquium, is really thanks to a group of faculty that last fall into spring, actually it was last fall, came to me and said, we really want to inaugurate a series where we would feature primarily some of the work and ideas of our own ESU faculty. And they launched a six-session series with Spring 17. It was such a hit, and I was so impressed what they were able to do. We continued it for this fall, and we hope to continue it. We really wanted to get you out of the classroom, members of the community, other faculty, to engage what we hopefully do best, or we are learning to do best, intellectual dialogue with our colleagues. So I really want to welcome you and thank you for joining us today. The other thing is the particular faculty I want to mention, and I'm going to turn it back to Dr. Eugene Galperin of the Math Department. He is the co-chair, along with Cem Zentaroglu, in communication. Last year, Bonnie Green in psychology co-chaired it with Eugene, and in fact, they were very key people for launching it, as I said. The other people are John Elwood in physics, Joni Bonatendi in art and design, Hushang Pazaki in sociology, Laura Pirangelli in nursing, Jan Selving in English, with great support from my assistant to the provost. Hi, good to see you. As well, Justin Amon, a recent graduate of ESU, and also Christina McDonald of the grants. So if anyone, one thing I love is to always quote Hillary Clinton, and that's not telling you my political bias, not much, but I really wanted to say, I always liked her phrase, it takes a village. And when I like to adapt it, it takes an academic village to make it happen. So if you would join me, and even though all my colleagues are not here that I just recognized, just give them a round of applause for putting this together. And if you don't think a provost, like looking at me, gets tired by this time, you would be wrong. I do get tired, but I have to say I have stayed for every one of the four so far, and I leave rejuvenated thanks to the intellectual discussions. You're on to a treat. Eugene Galperin will introduce them, but this is our little version. Maybe it will be televised on Black Mirror sometime. So I want to see the dialogue of science and philosophy on an interesting topic of genome editing. Eugene?
Well, thank you very much, Provost Bruno. I will be very brief. I don't want to stand in the way of most interesting presentation. I will just read the bios of the speakers. Dr. Kitchens-Kintz is the current Associate Dean of the College of Arts and Sciences, and her field of study is in molecular biology and genetics. Some of her research at ESU has included studying the effects of commercial green tea on human colon cancer cells and identifying genetically modified foods labeled as FDA certified organic. Dr. Connolly is an associate professor of philosophy and chair of the philosophy department. His major fields of study are ancient Greek philosophy, Chinese philosophy, and comparative philosophy. He has written and published 2 books entitled Foundations of Confucian Ethics and Doing Philosophy Comparatively, and has authored several published articles in his field. So without further ado, Dr. Kitchens-Kintz is going to start.
All right. Oh, there it is. Whew, came up. There we go. Okay, now what we're going to do today, Dr. Connolly and I are going to share this talk, and what my part is I'm going to try to teach people about DNA, about proteins, how mutations can affect the proteins, why we try to select for these things, and how we are making these mutations now. All right? Once I'm done, and don't worry, you know, this isn't going, you won't have to take a test on this, all right? And I love to see a lot of people who are not in science here now, all right? But once I'm done, I'm sort of giving you the tools with which then, when Dr. Connolly comes up, we can discuss kind of the good, bad, and ugly of this, all right? Is it right to do it? What should we do with it, all right? But you got to have an understanding of what's going on with that DNA, all right? And what we've been doing with DNA for over 6,000 years, all right? 6,000 years, all right? So again, my talk is going to consist of telling you about DNA and proteins. Again, the effects of the mutations on those genes, artificial selection, and this is us trying to bring out those mutations in genes, all right? Then we're talking about how we're actually making those changes through something called recombinant technologies, how it's being applied to gene therapy, and something called gene augmentation or enhancement, and then the latest and greatest way of changing the DNA called CRISPR, okay? Now, the first thing we have to know about are proteins. What are those things? Those things are the stuff of life. That's what we are made of, proteins, all right? Proteins do everything for us. And proteins are made of, if I can get this, of amino acids that are linked in a chain. And once they're linked in the chain, they fold up to form the protein, then the protein goes on to do its job. But now, what do proteins do for us? Like I said, everything. And what's amazing here too is that these amino acids here, there's only about 20 of them in life. It's only about 20 of them, all right? So life really is made up of 20 amino acids. Sorry about this, I have never used this before. Okay, so let me show. Whoops. Okay, now we're on track. Sorry about that. Now, what do proteins do for us? All right, like I said, almost everything. All right, they make our skin and bones. They transport things in and out of our cells. They make our hormones. They do reactions in our cells. They are antibodies, all right, that protect us, all right? So they just are doing everything. All right, now, the various, the various amino acids, in order for the protein to work, they have to be in a very defined sequence. very defined one. So these 20 amino acids for a protein to work has to be in a very defined sequence. If the sequence is off, let's say this histidine changes to a phenylalanine, it could potentially destroy the function of that protein, all right? Or it may not do anything to it, or it could actually improve the protein. So it can do three things. So proteins are very important, all right? Now, proteins themselves in the cell and in the organism's life, it's constantly being made, used, and then broken down again, all right? Constantly, all right? Now, some proteins will only be made and used for only 5 to 7 hours of the cell's life, because that's all that cell needs it for. Then once its use is done, the cell breaks it down. Some proteins hang out in us for years. But eventually, most proteins are going to be used, built, used, then broken down. And when the cell needs it again, it builds it again. So how does the cell know what the sequence of amino acids are for that particular protein? How does it know to build it in that way? Does anybody know? Science people? Go ahead. DNA. All right, it's DNA. DNA holds the blueprint that's going to tell the cell how to build that protein. That's what it's going to do. All right, so that's what the gene does, or the DNA. All right, so here we have the cell, we have the chromosomes, and then there's the DNA. Now, a gene, what is a gene? A gene is an area in the DNA that will tell the cell how to build that protein. All right? Now, so that's what a gene is. Oops, nuts. That's what a gene is. How does it do that? How does it do that? All right? It does it in two ways. Through something called nuts. I guess my thumb is really fat here, okay? So it does it through, here's the DNA, here's the gene within that DNA, and what's going to take place is that the cell is going to transcribe this gene. In other words, it's going to make a copy of that DNA. So it's going to make a copy of it. And it's going to use RNA instead of DNA. And then once that copy of the DNA is made, the cell then will read that RNA, and that's what then tells the cell what amino acid to put first, second, third, and fourth as it reads this thing. All right? So that's basically how it works. And let's kind of break that down just a little bit more. But in that process is called translation, because you are translating that RNA. All right? So let's, for instance, let's take this piece of DNA here. I just made this gene up. I just put some A, T, G's, and C's, because those are the bases that DNA is made of. And this area is a gene. And what the cell then will do is transcribe this by making a copy of one of the strands. And I just took this top strand, and I made a copy of it. Now, the copy is in RNA. Can anyone tell me the difference between this strand and this strand as far as bases are concerned? What's going on there?
You don't have a T, instead you have a U.
Right, so in the copy of the RNA, wherever there's a T in the DNA, the copy will, what they will use is a U base. But it's still transmitting the same information. All right, so now we've transcribed the gene, and here's the RNA, and then the cell will take this RNA and start translating it. And how it does that is that every 3 bases here, when it reads it, AUG, the cell will place the amino acid methionine down. All right, and then it reads the next 3. All right, the next 3 is CAU. And when it reads that, it knows to put the amino acid histidine there. Then it reads the next 3. All right? The next 3, AAA. The cell knows to put lysine there. This is known as the genetic code. This is the genetic code, all right? That's what everybody talks about, the code. Well, this is it. So when the cell sees AUG, methionine amino acid goes in that place, all right? CAU, in this case, histidine goes in this place. If there's any biochemist in here, I just made these up, all right? Now, except for AUG, because we all in science know that all proteins begin with methionine, all right? Now, after it's done, after it reads this and puts all the amino acids, it folds up, the protein then goes to do its job. And once it's done, it's broken down, all right? And then the cell, if it needs it again, rebuilds it by coming back to that gene, making a copy of it into the RNA, and then translating that. So it can do this again and again. All right? And what's so neat about this is that when does this gene do this? Some genes will be doing this only at certain points of your life, such as in embryogenesis. There are only certain genes that will be producing proteins involved in that. But once embryogenesis is done, those genes shut down supposedly forever. But they've now found that in cancer, some of those genes that should now be quiet are turning back on and making proteins that are making cells move. All right? I just think that's neat. That's the neatest part here, is when does that cell know to turn that gene on or not? Now, I hope you guys have gotten a good basic understanding of what DNA is doing. All right? What DNA is doing to make the proteins that we need. Okay? All right. Now, mutations in the DNA. They're happening all the time. All the time. They can happen inside the gene or outside the gene. If they happen within the inside of the gene, right here, that can potentially change the amino acid sequence of that protein. And again, what could then happen to the function of that protein? What can it do? Non-science people, Dr. Ball, what can it do? All right, if it, like I said in first, if that amino acid sequence is not correct, then the protein will either not function, it may not do anything to it, or it can improve it. All right? But that mutation is going to be transmitted to the protein. Let's take another look at that. Same gene that we had before, but look here. We've got a C here instead of the A. There's been a mutation. This could be caused by sunlight, by what we eat, or just luck, all right? Or just luck. But when this change does take place, when the cell copies that gene into RNA, you can see that C now is transmitted into the RNA. So when the cell starts to read that RNA, it gets the AUG, methionine, CAU, histidine, but now it goes AAC. It's not AAA anymore. It's AAC. And the cell says, oh, if I see AAC, I have to place an alanine there, not a lysine. All right, so now we've got a mutant protein now. Again, it can destroy the function of the protein, not do anything, or improve it. All right? Now, again, mutations are happening all the time. And mankind understood this, all right, that traits were passed on. They didn't know how, but they knew that these traits were passed on from one offspring to another. And if it was a trait that benefited human beings, they would try to pull that trait out, in this case, that mutation out to benefit them, all right? Not necessarily the organism itself, but to benefit them, all right? And this is known as artificial selection versus natural selection, all right? Now, over 6,000 years from the wolf, because all dogs did descend from the wolf, this is the kind of mutations that we have from this wolf, all right? Now, let me ask you a question. Do you think the mutations that made this little dog is good for that particular dog? Could they live in the wild? No, They couldn't. So these mutations really benefited the humans and not the organism itself. But we've been doing this for 6,000 years, not only to our animals, but to our plants as well. So take a look at this little plant here. This is how corn started out. Look what we have today. All right? But again, these mutations take time to happen. They have to happen in the right place, all right, and give us the right trait. So this has been 6,000 years that we have been genetically modifying our organisms. So in other words, we've been eating GMOs for 6,000 years. What's the difference from then to today is that back then, and even still now, we had to wait for Mother Nature to make the change. Today, we are trying to make the change, all right? Because around the 1940s, they finally learned it was the gene and the DNA and the gene within that DNA that was causing these traits, and mutations as well would change these traits, all right? So we figured that out. And we said, you know, we want to speed this mutation rate up. So what they did was they took, usually they had to do it on single cells. They would take bacteria, put it into a chemical mutagen, and then of course this chemical was just changing the DNA in these cells, all right? And then they would try to select then after a while for the bacteria that they wanted, all right? So we're speeding up the mutation rate. We're doing it now, okay? But again, you cannot do this on a horse or a cow or a pig or anything like that. But it was in the 1970s that they discovered these DNA or these bacterial proteins that cut DNA at very defined spots. And with that kind of power, people started then, they found out they could cut the genomes of organisms, all right, from one organism and then another, and then recombine them. And now what you have is a man-made genome. A man-made genome. This is why it's called recombinant technologies. You're recombining DNA. In this case, they've taken a growth hormone from the human, put it into a bacterial gene, and then took that piece of DNA and placed it back into the cell. And that bacterial cell will transcribe and translate the human growth protein. And why? Does anybody know why? Why would we bother doing this? Well, what's the source for human growth protein prior to this technology? Humans, all right? You didn't get a lot of it. But now, with this, they can generate lots of this stuff, all right? Lots of this stuff. Insulin is being done in the same way. You could also add genes into the genome of other organisms and giving them functions they've never seen before, okay? In this case, what we have right now is a pig that's being humanized, all right, humanized by placing human genes into the pig. Why on earth do we do this? Because pigs are a great source of organs for us. They look almost exactly like ours. What's a major drawback of organ transplant right now? Supply. Supply. People are dying, all right? But when you put a pig organ, what happens? You reject it severely. So they're trying to reduce that by humanizing the pig, all right? They also now have a goat that produces silk in their milk. They have it, and it's being used now, all right? And so they got some spider genes in there, all right, in this goat. Now, why would you do that? Well, we want a big source of silk, and these little animals that produce it are only this big. But now we have a goat, has a huge milk bag, and now we can get lots of silk now, okay? Lots and lots of silk. And what they're trying to do as well is to get these corn ears or some corn plants to produce medicines for us. That's still kind of far away, all right? Other things they're doing, which is pretty neat, and we have today, pineapples that contain carrot genes to increase the nutrition of these things. We have little animals now, most of our pets, cats, dogs, pigs, horses, cows. They've taken this jellyfish gene that makes the jellyfish glow green and place it into these animals. And so when you shine a black light on them, they glow green. All right? They're doing this for developmental studies, but somebody's got the idea, we can sell these things. Designer pets, and they are selling the fish that way. And speaking of fish, and here they've actually put a growth hormone gene in here. These two fish are of the same age, but one is growing 3 times faster than the other. Okay? That's here now. All right? But with these techniques, there's lots of problems. All right? Lots of them. All right? First off, where are the genes going to land in the genome? All right? You're going to stick this gene in there, but we can't really control where it lands. And where it does land is very important because it can make the organism less fit. And in fact, they've now shown that there's a GMO soybean plant that is resistant to the herbicide Roundup. All right? And it's a bacterial gene that they put into this plant. They have found that these GMO soybean plants are not as fit as the regular soybean plant. All right? Their roots don't grow as deep. All right? Other problems affecting non-target organisms. There's a corn plant that makes its own pesticide. And that's good when you want to kill the insects that eat the corn plant. But the monarch butterfly eats the pollen. These genes are also in there as well, and it's killing them. All right? So it's a non-target organism. Also, unwanted artificial selection. As we spray these fields with herbicide and these GMO soybeans can resist it, the natural weed plants are naturally evolving to resist the Roundup. So we're going to be back at square one. Okay? And then another one is controlling where these genes move around the world. Because right now, the GMOs are contaminating our food, and nobody can control it. Monsanto had a wheat field, a GMO wheat field, and there was such a backlash against this, they took the wheat plant off. All right? They said, okay, we'll take it off. This was 10 years ago in Montana. Last year, those GMO genes showed up in the wheat in Oregon. All right? So these things are spreading, so you can't control them. Now, another thing with recombinant technology is gene therapy. In this case, we're trying to insert a normal gene to compensate for a bad gene. That's what we're trying to do. But again, lots of problems with that. And in fact, we've been doing this for 30 years, and we haven't gotten really past clinical phase 2. All right? So it's just riddled with problems in here, such that you have to modify the cells outside the body. When you put them back in, they just disappear. Or you have to use a virus if you want to modify inside. And you have to modify the virus itself, and sometimes it can revert back to its virulent form. All right? So there's all kinds of problems with this. Now, today, though, we have CRISPR. All right? Now, CRISPR has been known for about 30 years, maybe 20 years. And it's a natural system found in bacterial cells, a collection of proteins that actually work to protect the bacterial cell from a virus attack, because they are attacked by those. All right? But only present but future attacks. And in fact, they think this is where it's the first instance of acquired immunity on Earth. All right? Now, someone about 10 years ago decided they're going to modify that CRISPR system such that it can go in any place you want it to. It is extremely highly specific. Anywhere you want it to, and change the DNA to exactly what you want. All right? So now we can actually go in, and instead of inserting a good gene, we can go to the bad gene and correct it. All right? Correct it. We don't have to add a gene. Now, CRISPR still does that. It still can add genes in, but now you can pick the exact spot where you want to go. I want to put it right there. All right? The techniques that we had, he was like, you put it in, you go, where it lands, I have no idea. All right? But now with CRISPR, I can say, I want to put it right there. All right? Or I want to correct the gene, not add one in. Not only that, the techniques that I just talked about are laborious, and they cost a lot, a lot. It took me 2 years to make a mouse that was carrying a human gene. all right? 2 years, all right? You can do this in a matter of days or weeks. And the cost is low. It's low. And in fact, someone told me, I think in South Korea, they're selling CRISPR kits now. I don't know about that. All right, I'll have to go and research that. But CRISPR is easy and it's low cost, all right, and it's more specific than what we've ever seen, all right? Now, right now, CRISPR is being used towards gene therapy, but with it being so low cost, people are now looking to use it for gene augmentation or enhancement. And what is enhancement? Well, in gene therapy, you go from not normal to the norm. With enhancement, you're going from norm to beyond. All right? That's what enhancement is. All right? Well, what kind of things do we want to enhance? Well, our intelligence. All right? We want to be stronger. We want to be more athletic. We want our offspring to be the same way. All right? But what is the norm for human beings? All right? And this is what Dr. Connolly now is going to talk about. What's norm today was not norm 30 years ago or 100 years ago. So norm is continually changing. And with that, all right, should we be excited about this new CRISPR or totally frightened? All right? Now I said, well, breaking news, because this was yesterday. People in China just CRISPR'd 12 pigs. All right? And what they did was that they put this mouse gene in there, UPC1, from a mouse. And what it does is it takes the pig's, 24% of the pig's fat, and shunts it towards the production of heat, all right, to make basically a hotter pig, all right? And why? It costs a lot of money to keep the pigs warm in winter. You got a hotter pig, you don't have to spend as much to keep them warm. All right? And it makes them skinnier as well. I'm like, well, that's cool. But what happens to these pigs in the summer? Are they going to suffer? You know, I don't know. And what about that taste? You know? Because, like, I know what I am after when I eat the bacon and ham. And believe me, most of that's the fat that I'm after. And now I'm getting a skinny pig? Heck no, I don't want a skinny pig. All right? Now, given that, it is Dr. Connolly's turn now to talk about, now, hopefully I've given you the tools now to discuss the good, bad, and ugly of this. All right.
I'm thinking about becoming a vegetarian after hearing that. My name is Tim Connolly. I'm from the philosophy department. I'm here to talk about the ethical implications of CRISPR. I've also brought with me a student in our department, William Collier, who's going to talk about the political and social implications of CRISPR. And I think we don't just want to talk about it, but we want to have a debate and discussion, right? So this is the audience involvement portion of the program. Also, I'd like to thank Dr. Kitchens-Kintz for inviting me to be part of this presentation. I think it will be a lot of fun, and I've really enjoyed learning about CRISPR from her. How soon is the future?
The future is now.
Okay, we can do it now, as the cover on the one magazine says. We can do it now, and this is actually a couple of years old, right? And then they had a Time magazine cover story that said 2045 is the year we all become immortal. I hope I make it that long, right? Or maybe I don't hope I make it that long. But one thing I've learned from listening to you, Professor, is actually a lot of this stuff is already in the past, right? So in the 1990s, long time ago, they had gene therapy. And in the 70s, they had these recombinant technologies. And then the history goes back 6,000 years if you're talking about artificial selection. So all of this stuff has been happening. 2017, I think it's going to be a year to remember for a lot of reasons, right? But one reason it's going to be memorable is the first human embryos were edited in the United States. And this happened about 3 months ago at a university in Oregon. How many of you knew this already? I'm just curious. Right. Okay, not too many of you, not too many of you. So this is an important part of scientific history, I think. Probably because the technology is moving so rapidly and public opinion changes so slowly, I think public opinion has not gotten caught up with the changes that are happening. So if you look at the one graph, it says there's more worry than enthusiasm. And this is just about gene editing that is therapeutic in nature, right? So not to give the organism any enhancement, right? But just therapeutic. Most people are worried, right? Fewer people are enthusiastic. And then I think this research survey is very interesting too. 42% of Americans have heard of CRISPR, not at all. 48% a little. That means 90% of Americans know nothing or just a little about this kind of technology. So the public opinion lags far behind. I'm just curious, how many of you have heard of CRISPR before? All right. How many of you have studied it in one of your courses? All right. What course? You guys have the shirts. Where do I get one of those shirts?
The Biotech and Medical Tech Club.
Oh, the Biotech Club. Awesome. Awesome. Okay, great. Great. I knew I could count on you guys. Okay, give me a form. Give me a form. All right. It didn't help last year when the director of national intelligence said CRISPR was a weapon of mass destruction. And on the national threat assessment for 2016, they had things like ballistic missiles, long-range ballistic missiles in North Korea, chemical weapons in Iraq and Syria, and CRISPR, right? So did you know that you were studying a weapon of mass destruction? Or at least so says our recent threat assessment. And at first you think this is a crazy thing to say, but then you consider that it's low cost, right? And that it's distributed among many different countries, right? Many different people. Yes, the thing in South Korea that you mentioned, I didn't know about that.
I don't know if it's true or not.
Okay, but it's something to be worried about. Here are a few things philosophy can contribute. I got to make a plug for philosophy before we move any further. So we train our students in analysis to break down the deeper meaning of the main concepts that are involved in these issues, concepts like human nature, right? We spend a lot of time talking about. We train our students in ethical reasoning to discuss the main moral principles that are involved in these debates. We train our students in logic. There's Professor Priam over there who teaches logic in our department. Okay, so we train our students very seriously in the ability to recognize good arguments and bad arguments. And I think there are good arguments in favor of human enhancement, and there are bad arguments in favor of human enhancement. And if you stick to biology courses, maybe you don't get that sort of logical training, right? So I think philosophy can help a little bit. We encourage debate and discussion. We want our students to ask the big questions. In fall 2018, we are offering a bioethics course for the first time, right? 2018 is going to be another banner year, right? So thank you, thank you. So we hope we can attract not just students who are interested in philosophy, but students who are interested in biotechnology and other hard sciences, but also students from political science and sociology and psychology and all the kinds of disciplines that are relevant to these questions. So I think what's special about this kind of debate is the issue involves so many different fields, right? So it should be a collaborative discussion between all of us about what happens. All right, let's get to the questions. Let's get to the questions. I think the first and most obvious question, and the most specific one, is which uses of genome editing are morally permissible, right? So now you get to vote. Everyone has to vote. Okay, I'm going to be looking around. I'm going to be looking around. You have to say yes or no. I suppose you could edit your child's genome so that instead of having the chance of living to 28, they would have the chance of living to the age of 78. Would you do it? Right? Think about it for a moment. All right, how many votes for yes? Don't be shy. Don't be shy. All right, any votes for no? I'm just curious. Okay, much fewer votes for no. Much fewer votes for no, but I think I saw about 90% votes for yes. If you take one of my philosophy courses, you learn it's always the second question that matters. And the first question is just a setup, all right? Suppose you could edit your child's genome so that instead of living to 78, they would live to the age of 128. Would you do it? Right? Think about it for a moment. Think about it for a moment. All right, I'm actually curious. Anybody want to make a prediction on the ratio of yes to no? 50-50. Okay. Okay, think about it carefully. And I'm going to take down names, just so I know who to worry about. Right? Okay, how many votes? No questions. No questions, sir. How many votes? Whatever condition it will be, right? Whatever. And also, why didn't you ask that with the first question? Because it's very relevant. Yeah, but I think it's relevant to both, to the consideration of both questions, but maybe more relevant to the second one. Way more relevant. Yes, way more relevant. All right, how many votes for yes in this scenario? Okay, how many votes for no in this scenario? All right, so mostly no's, mostly no's. How many of you changed your mind from scenario 1 to scenario 2? Right? Can I ask, who wants to answer, right? Why did you change your mind?
I think in the first case scenario, we have some sort of expectation of what we think is going to happen in the long term, whereas in the second scenario, no one could say for certain until 128 years have passed.
All right, it's unknown. It's unknown. Yeah. All right, and that's the question about quality of life. All right, anybody else change your mind for a different reason? Why did you change your mind?
It's the quality of life issue.
The quality of life issue.
Because I know what happens after 78.
But suppose we could just average out the quality of life over 128 years, right? So whatever arc you would have from birth to age 78, we just transpose that arc onto birth to 128. Would you say yes?
Oh, yeah.
Okay, interesting. This gets at one of the most crucial distinctions in the ethical discussion of genome editing, and it's the distinction between intervention and enhancement. Intervention takes the organism up to normal human functioning, right? Or it gives the organism the chance at normal human functioning. And enhancement gives the organism the ability to climb walls, right? And do the sorts of things Spider-Man can do, right? So there's a difference between intervention and enhancement. I think it's possible the answers to these questions are getting at this distinction, right? In the first case, that might be classified as an intervention. In the second case, we're talking about an enhancement, right? So we might think which uses of the genome are morally permissible. The ones that intervene to bring the organism up to normal expectations are permissible. The ones that don't are not. The ones that go beyond normal are not permitted. Okay, what's your question, sir? All right, it's a good question. You will do well in philosophy, right, if you're asking that question. So, unfortunately, some people say the intervention versus enhancement distinction does not make sense. And I think they've actually got some good reasons to say it does not make sense. This fellow right here is Nick Bostrom. He's the director of the Center for Future Studies at Oxford University. And he says, actually, this distinction collapses upon examination. So for instance, is vaccination an intervention or an enhancement? I mean, in that case, are we intervening to bring the organism up to normal capacities, or are we enhancing it and giving it a sort of immunity that it would not otherwise have? What do you say?
I would say it would be more of an intervention, because due to vaccinations, a lot of the diseases that would kill a person, a child, really young age, now you are allowing them to at least live longer, which will be by standards of regular life.
Yes, but this proves the point, right? Because an intervention becomes a kind of enhancement that then we consider to be the norm, right? So it's very hard to separate these two, right? All right, anybody else want to weigh in on this? Is vaccination intervention or enhancement? I mean, vaccination should be permitted, right? Okay, that's a separate thing. But most people would say that's an acceptable kind of intervention in the life of a biological organism, right? You stole my question. All right. how to define a normal state, right? The normal states are always changing, right? So you can think about human nature, right? Human life expectancy in the Stone Age was between 20 to 34 years. Right? So that's Stone Age normal. In the past 150 years, life expectancy in most advanced nations has increased at the rate of 2.5 years per decade. Right? Think about that. So normal 150 years ago for lifespan is not normal now. And even normal 10 or 20 years ago is not normal. And then the normal in countries with the most advanced medical technology is not the norm in countries without them. Right? So what is our natural lifespan? Yes?
It's always changing.
All right, so there's no natural lifespan.
You keep intervening.
All right, one thing I'm curious for the biologists in the audience, how do you address this question of human nature? Because it seems like human nature is a very relevant concept to thinking about the ethics of enhancement, right? But if what's natural or normal is always changing, right, what do we do with this concept?
Don't know, because now they find that, I read an article years ago that said aging was a new phenomenon upon the earth. It's new. So we're kind of going into a new world, all right, as we age. Because most people would die at their age of 20 to 34 years of age. But we're expanding that, So aging is new. It's new to this earth.
All right, it's not, is it natural or not natural?
That's a debate. I would say natural if you intervene.
Okay, so you see, these distinctions are hard to track, right? Yes, yes, please.
For natural age, for a human and all animals are controlled by a link in area in DNA we call it telomeres. No matter what, 100 years ago or 1,000 years ago, it will be the same. However, the environment changed, that's how we change our natural age. But naturally, look at the cat, dog, the human. If you live normally, you should have certain age. After that, you have to die, naturally. So it doesn't change just from decade to decade. You might live longer or shorter, but your natural age controlled by an area in the DNA, we call it telomeres.
Right, so how does that help us? How does that help us with this question?
That means you can improve or elongate the lifespan or something. That's because the environment changed, the healthcare changed, things like this. But naturally, we don't change the telomeres in our DNA.
Yeah, so when we're talking about is something an intervention or an enhancement, that still doesn't tell us which is which, right?
If you talk about vaccination and all this healthcare, we call it enhancement. Because naturally your body have defense mechanisms. Your body, your immune system make own vaccination. Just we try to manipulate that system to give us much better protection, longer, easier. That's what it is. It's just only enhancement. Your body is controlled by your DNA, by what you get in your body.
Yes, so do you think this distinction is a meaningful distinction? This one. Is this a meaningful divide?
I go for enhancement. Just what we do, everything we do is just enhancement.
Yeah. All right, let's hear a few more. Yeah, in the back.
I just want to point out that the two things that have made probably the largest difference in human lifetime are not any medical per se, but it's called soap and it's called sanitation. Those are two huge things that have changed our lifespan. And the reason we died when we were 35 was because we got sick from dysentery, or we got sick from bad food. And those things are much less prevalent now because we know, well, wash your hands, wash your food. Make sure your outhouse is on the downhill side from your well. Those are basic things that we now understand. understand.
All right, let's hear a few more in the back, and then we'll move over here. Yeah.
Can we possibly make a distinction between prevention and enhancement?
I'm sorry?
Can we possibly make a distinction between enhancement and prevention?
Yes, I think that's what this distinction is trying to capture, right? So one way of thinking about intervention is it's an intervention to prevent some sort of inherited disease that would otherwise cut the organism's lifespan short. So usually it's preventative intervention, and the one that they did already way back here, this one, right? That's what they were trying to do. Okay, over here. Yeah.
So the distinction, as far as I can see it, is relative.
Yeah, I think that's what these comments suggest, right? As many people might think this is an intuitive distinction, right, as captured by our answers to these questions, right? The one is an intervention that brings the organism up to the norm. The other is an enhancement, right? But if you say it's all enhancement, then that doesn't really work as a distinction. So we need some ethical basis for saying which genome editing is permissible. And some commentators like Bostrom suggest that that distinction doesn't do it, right? So if anybody has an alternative distinction for telling us which genome editing is permissible, which is not permissible, then we can entertain that suggestion. Yes?
I think, don't we want to avoid a situation where you have a certain class of people who, because of their wealth, can afford to create children who are intellectually superior?
We're going to talk about this, I promise. Yeah, and I think that's part of the reason for worry. We want to avoid that sort of situation, right? So you think about the social implications of enhancement, and it becomes problematic. Jamie.
Back to the distinction between natural and placing where is the norm, where is the natural. You could also make the argument that it's natural for people to pursue betterment. So from one perspective, I naturally am inclined to find food. Why shouldn't I be naturally inclined toward changing my DNA?
Yeah, that's why we have to address the question of nature. Is anything about us natural? Some philosophers, beginning with Giovanni Pico della Mirandola, who was a 15th century Italian thinker, say what's natural about us is just that we change our nature. Right? That's the only natural thing. So we are the unfinished animal. Right? In other species, they have a limit, right, to what they can attain, but the human being has no limits. In philosophy, we raise some difficulties with the concept of nature. There's something called a naturalistic fallacy, right? Just because something is natural doesn't mean it's moral. Just because something is unnatural doesn't mean it's immoral, right? So things like typhoons and earthquakes and early death from wasting disease, those might all be found in nature, but we don't think they're good and we try to prevent them. And also something like Tylenol, right, or vaccination, those things are human, artificial inventions, but we think they're good. They're for the better. So natural doesn't mean good, unnatural doesn't mean bad. There's also something in psychology called the end of history bias. Right? The end of history bias is that we're living at the end of history, and how we are now is how we are meant to be. Right? So the development that's occurred for human beings is all in the past. And we're the product of that development. But we don't think, no, we're part of the process of development. So in the literature about genome editing, there are these two positions. Bioconservatism is at one end of the spectrum. And it says we should be worried, right? Remember, worried or excited, right? Bioconservatism says we should be worried, right? And we should adopt our social policies so that we limit this kind of experimentation, or we at least set a high bar. Right? So you have to fill out a lot more forms than you already do if you want to do this kind of research. Right? And the bioconservatives have interesting arguments. And they're not just people who are politically conservative, by the way. Right? Like some of them say enhancement is a threat to human dignity, and that might include many religious people. Right? But not just religious people. Some philosophers who are secular philosophers have said it's going to change the parent-child relationship, right? If you have parents choosing for their children what sorts of traits they ought to have, that violates the child's autonomy. Do you guys know this word, autonomy? It's the most important word we teach in philosophy. What does that word mean?
Free will?
Yeah, the ability to choose for oneself, right? So we would say certain interventions prohibit or cut off the ability to choose for oneself, right? And if parents are meddling in their children's genetic traits, right, that would be a violation of autonomy. And what matters in ethics is that we protect autonomy for each person. We protect the ability to choose. It will lead to social inequalities. This is something William's going to talk about a little bit more. And so, some of the people in this camp are actually environmentalists. They think technology has already led to massive degradation of our natural environments, right? And if we turn it on ourselves, it's going to lead to even further devastation. So they say, be skeptical, be wary. All right, at the other extreme, you have transhumanism, right? Have you guys studied transhumanism before? So it says we should embrace these things. We should embrace these sorts of changes. All technology is a kind of enhancement. In general, technology has made our lives much better, right? It's undeniable. It's undeniable. Soap, right? If you think about soap, right, and things like that, it has made our lives better, right? Okay, and we're not even talking about what more advanced things like Nintendo and okay, all right, I'm old too, right? And gene editing is going to make our lives even better than previous technologies, right? So our lives will be longer lasting. They will be more pleasurable, right? We will have greater creative capacities. We will have more reproductive choice, right? Okay, so let me ask, which side of the spectrum do you lean closer towards? Or if you want to carve out a middle position between these two, like feel free to do it. But do you find yourself leaning closer towards the bioconservative end of the spectrum or the transhumanist end of the spectrum? Just if you had to pick one, which one would you pick? Right. How many votes for bioconservatism? Don't be shy, okay? It's okay. Interesting. I count about half of you. How many votes for transhumanism? All right, what a productive discussion this has been. All right, so you see, smart people are split on this issue, and that's why we need to talk about it. That's why we need to talk about it. That's why we need to get it out there. And we need to make sure, at the very least, the public has information, right? And the public knows what is going on, right? So I think that's why we need at least a little philosophy to get the discussion going, to stake out the various kinds of arguments and the positions we might take, lay out the concepts. Also, we're fun to argue with. Okay, we're fun to argue with. All right, thank you very much. Thank you very much. And I will now introduce William, who is a philosophy minor and who is going to say a few things about the social implications of CRISPR technology.
I'm actually going to start off with a YouTube video, break the norm of presentation. It's from a movie.
No damn well why. Back off.
Can you put the captions on, please?
Yes.
Oh, I'm starting to want you to make me. Yeah. Big man in a suit of armor. Take that off. What are you?
Genius, billionaire, playboy, philanthropist.
I know guys with none of that worth 10 of you. I've seen the footage. The only thing you really fight for is yourself. You're not the guy to make the sacrifice play, to lay down on a wire and let the other guy crawl over you.
I think I would just cut the wire.
Always a way out. You know, you may not be a threat, but you better stop pretending to be a hero.
A hero? Like you? You're a laboratory experiment, Rogers. Everything special about you came out of a bottle.
So, Captain America owes his strength, agility, all of his powers to the serum he was given earlier in the storyline. And as genetic engineering becomes more advanced, we are going to see more people like Captain America emerge And with that is going to come a wave of skeptics who will try to undermine the accomplishments of enhanced individuals. How many people here play a sport or have ever played a sport? Now, what sport have you played?
Ultimate Frisbee.
Ultimate Frisbee. How would you feel if the people you're playing on the other team were all enhanced? They were all genetically engineered. They're all a foot taller than you. They all have twice the muscle mass. They're all objectively smarter than you. How would that feel?
I mean, it wouldn't be fun, but most of those people are already taller and stronger.
Now, for other sports like track, football, baseball, sports that tend to have records, imagine that these people start to break all the records by miles. They're able to run 4-minute miles, barely breaking a sweat. What will that mean for the social interactions? Should we put asterisks on their names when they start to break the records? Should we treat them differently if they're breaking barriers in sports and making accomplishments? And the problem with that is once we start to see them as differently and treat their accomplishments and undermine their accomplishments to say, it will separate us. It'll say that the genetically engineered are different from us, the naturals. And the consequences that can have are very drastic. We already see many people as different based on race and gender, sex, but now it'll be because of their height or because they have full heads of hair. And I think a larger part of this problem is that this will not just be because of the technology, but it will be because of access to technology. The rich or the generally wealthy will be able to do this freely, and the poor or people who don't have access to technology in other countries where the labs aren't there will not, and they will not be able to enhance themselves. And even more so, for all of us that are able to get this technology, the traits in the countries where they're at will become more prevalent. In America, most sperm banks ask that you be around 5'9" or taller if you want to donate sperm. So anyone shorter than that in the future, when all of these qualities become more prevalent, will be seen as a sort of outcast. Full heads of hair will become more common, and anyone that doesn't have that will be also seen as an outcast. And so we have to ask ourselves, do we want to have a world where everyone is able to freely give themselves all these enhancements and make themselves into people like Captain America, or should we put a cap on it and say, no enhancements, no vanity traits, nothing to improve ourselves, as Professor Connolly was saying, beyond subjectively what is normal? Or should we create a baseline saying only things that bring people to healthy living, getting rid of genetic diseases, getting rid of any ailments, disabilities, however you put it? What should we do with limiting genetic engineering? That's it.
All right, now I think we can have free-ranging discussion, right? So if you want to ask
questions.
a question to anybody, or if you just want to say something, right? I think now would be the time.
Dr. Hunt's had his hand up over here.
Oh,
I'm
okay,
sorry.
okay,
So
you call.
Peter's
you call.
next.
Yeah.
Dr. Hunt.
So I wanted to make a point and then kind of 2 points, and then I'll just leave it for discussion. One is, it's not true that all animals are destined to die. There are animals that continue to make telomerase throughout their lives, and if they weren't eaten, stepped on, or squished, like the hydra, like certain jellyfish, like the lobster, could theoretically live forever. We've got lobsters that are over 400 years old. We have coral that's over 1,000 years old. So I think that becomes part of the discussion. We should not just assume that we have an upper limit. We may because of our complexity as vertebrates, but we may not. The second thing I want to put forward is probably not a unique idea in the room is Rich people are enhancing the performance of their children today without genetics. We're able to hire tutors, to give them the best nutrition, to give them the environments in which they can excel and be the best athletes, be the best students. I'm not sure I see the ethical moral distinction between that and this.
This might just be greater degree, right, but not a different kind. Yeah, thank you for these points. It's wonderful. Yeah.
Dr. Brian.
I think it might be illuminating to draw an analogy between how might, what's it called again? What's the, CRISPR?
CRISPR.
How CRISPR might change life compared to how has the cell phone changed life. That is, there are many analogs there in that I don't think it's a question of technology, yes or no, good or bad, but it does teach us things like the question is more complicated than saying natural is better or more is better. I think, for example, what technology has done to our love of music. Used to be it's hard to get music, you had to get your LPs, you had a very carefully curated collection that you loved and knew. And now it's, I have access to 10,000 songs, so no one of them really matters the way it used to matter. So it may be that what we have to do is not look to the dimensions of natural, unnatural, more versus less, but rather something about what makes human life dignified, valuable, and precious. And does more of this or less of this help with that? Does more or less of this have with that? And so we might ask, if we say, indeed, human beings are so malleable, we have so many ways we can change, but what is it that makes us good? It's something about dignity, intelligence, empathy, community, and so we need to think deeper about that. Then we can figure out, now if I add this new piece with my technology, is it improving that or not? If we ignore the deeper philosophical question of what makes human life worth living, if we do that, then we're only talking about, I could have more of this, but to what end?
Sorry.
Personal story that relates the extent to which we are already on this slippery slope. I once dated a woman who was in med school who had, not surprisingly, a bunch of staggeringly insensitive friends. And so at a party one time, when they met me, they said, you know, in this day and age, no one would have to endure the tragedy of your life. And I asked what they meant. They said, well, you're 5'4". And that's a disease. And I said, define how that's a disease. And they said, well, you're in the bottom 2% of the growth chart. And that's how we define that you have a disease. And I said, so if I've been given human growth hormone, then that means that somebody else would have taken my place in the bottom 2%, and then I would have given them a disease. Because there's still going to be a bottom 2%. And if I'm not in it anymore, somebody else is in it. So that we could define disease as being a thing that if you fix one, somebody else has it.
You got to get some new friends.
These were not my friends. These were my then-girlfriend's friend. But it kind of ties into a lot of these questions. What do you call normal? And if you change what you call normal, what does that mean? And, you know, et cetera.
It's a very serious question. What do you call normal? How do you define normal? If we're using normal as a normative category, right, meaning it sets a norm, right? It gives us an ethical aspiration, or it sets an ethical standard. So if we're using it as a standard for ethics, then it's a problem, right? If we're just talking about it in terms of average, it's not a problem, right?
And for what it's worth, thinking back, I'm really glad nobody gave me human growth hormone. And I'm really glad that I didn't turn out to be more normal in the sense of what all med students were thinking. But in this day and age, parents are being told that's a tragedy that you have to fix. As in similar way with so many other diseases.
Yes, so do you see the concern about autonomy, right? It's that it should be up to you, right, to decide rather than to your parents to decide for you.
Or to the statisticians.
Oh,
We had somebody back here.
technology develops and, you know, we as a society decide that we should cap it, you know, you know, so are we depending on the government to create laws for this? And like, criminals don't follow laws, so I think it's, you know, probably going to happen either way, like in like underground laboratories or something, you know, the technology's there.
Well, it's kind of curious in that when you say criminals don't follow laws, so you don't necessarily have to be a criminal and not follow the law. Because we can set a law here, but in Canada, well, they say, well, we don't care about your law. It's not ours. And so we're going to go forward, all right? And then even if the world did set a law, how are you going to enforce it? Because that's what police are law enforcements. So how are you then going to enforce that law? So those are really big things that we have to look at. Because right now, I mean, you're on the road and you see people speeding by you, they're breaking the law. Where are those law enforcers? They're not around. I always say, where's the cop when you need them? All right? Because people are always breaking laws every day, okay?
We need a new major, like bio-criminal justice.
Yeah, so we got, well, hold on, Dr. Archer, we got a student here, and then you're next.
I was just going to say on the point about athletes, like, performing their own, or, like, trying to, like, increase, like, if you can be, you know, 6'8" and be faster and stuff. I mean, that's kind of why steroids are banned in the Olympics and stuff. Like,
exactly.
Why Lance Armstrong had, you know, some of these things taken away was because he cheated. So, I mean, I don't know how you can, you know, I guess it does get murkier to enforce, but I'm sure that, you know, if there were the guidelines, that you can't just genetically modify yourself to run a 3-minute mile and, you know, break the world record and then be like, well, I did it through hard work.
Yeah, William talked about, he and I talked about the same thing.
Yeah.
So, and what we were saying is that, how are you going to know? Because right now, you can test someone's blood for these enhancing drugs. But if you're CRISPR, all right, how are you going to know? How are you going to know? Unless you put a DNA tag in that person. And then you can go look for that tag, all right? So then are you going to require to have a tag if you CRISPR, all right? And again, who's going to enforce the law?
I was also going to say that the technology will get to the point when you're able to genetically modify embryos. We already saw it in the US. And it might get to the point where you can genetically modify your kid to get double the muscle development throughout their entire life. And then it's not their choice. And then they go on to be an athlete. It's not intentional. Whereas with steroids, I take steroids now. It's my own free will. I do it with my own intention. Whereas if my parents designed me to be that way, to be taller, it's not my call. And then if I'm then limited by getting an asterisk on my name my entire life, what's that going to mean for me? Any accomplishment, will it be mine?
Dr. Aldrich and Dr. Chow?
As the technology advanced at accelerated rate, I think this is the right time for the society to decide what we need to use it for. Otherwise, if we keep it open, we end up with disaster, like many things happened in the past. But the society has to still until all of us have understanding for these, when we can use it. I understand to use it to correct certain defective genes, fatality, save lives, save people suffering and pain. Everybody, this is legitimate use. Beyond this point, to increase your height, your intelligence, your color, this is misuse, abuse of the technology. And who can stop it? The society. Like society say, no cloning to humans. That's it. You put the point to stop it. Nobody, even any scientist in the world, they don't think about it. Some people they think, but they do it in their own lab. Okay? But that's it. Cut. No more human cloning. You cut. The same thing for CRISPR, and it's getting more advanced. Just today they found a new technology, they call it base pair, you can fix it. Even you don't need the whole CRISPR, you need just one base, you can change it from AT to GC. It's just amazing technology. So we need, the society has to put the limit. You can use it, yes, to correct certain defective gene, fatality, certain suffering pain, increase the productivity for certain product, increase, that's it. Not to increase your height, your color, your intelligence, this abuse. And that's how we stop it. Otherwise, it will be open for everybody, as Dr. Kitchens said, it's become available. Anybody in the lab can do it. I can purchase it today, just through email. They send me CRISPR, I can inject with these animal bacteria and can fix it. So we have to stop and define when we can use it, who can use it.
Dr. Tower.
One of the things that I think is really interesting is when people are making the distinction between organic enhancement versus what we already do. We have artificial types of organs that we do, our limbs, hips, so forth. And so we don't seem to have an issue with that. And a lot of times the argument is that, well, that individual not only makes the choice, but it dies with them. So when you talk about genetic enhancement, if you made the choice to genetically enhance yourself, would that be something that's okay versus if you use that technology to genetically engineer the next generation? So if you use CRISPR to then change the sex cells that you make for the next generation, you're taking that away, that choice. But we seem to be fine with people that make choices on themselves of enhancing. If, well, okay, I have a bad heart and I need to replace, okay, we talk about pain and suffering. Who defines the pain and suffering? That's one thing. But another thing I always think is interesting is that once you, like you said before, I'm also some molecular biologist for people that don't know me, but we like to make things, don't we? Okay, genetically engineer things. When the cat's out of the bag, so to speak, and the technology is out there, you can't put it back. Okay, and you talk about then trying to police things. Okay, well, if let's say someone were to do something, make genetically enhanced, highly intelligent human beings, if you punish that individual, fine, but now those human beings are here. Are you going to tell me you're not going to then try to get majority what you can out of their intellectual power? What if they come up with new technologies? A better iPhone. Are you going to say, oh, I'm not going to buy that because it came from people that shouldn't have been made? Well, come on. Okay, so I find this there's a lot of times when we get into these conversations, a very circular types of thing, but we always come up with excuses of when we wouldn't stop it though. Well, for that would be fine. And it's that slippery slope, of course, that we were talking about before. So I love these conversations, but the thing is that once you get the technology, it's there. How are you going to deal with it?
All right, you guys sit here.
I think it's irrational to ask a simple yes or no. I think that it involves such a broad array of genes. I think we should be comparing it to the pharmaceutical industry, which already produce lots of synthesized drugs or medications that augment our life. Perhaps we should look at it on a gene-by-gene basis, and perhaps the development. I mean, I don't think we fully understand what each and every gene does already. So perhaps, as we discover the effects on new genes, we could go through an FDA process for each individual gene that we look at, and similar to the way that we have drugs approved.
We have a student over here. Did you want to speak still?
I was just going to make a comment on there are certain advancements we've already made that are kind of comparable when they become accessible, we're more accepting of them. So, for instance, contacts or LASIK surgery in sports. You never question that, but it is a form of enhancement. So, once it's become the status quo, we kind of forget about it.
All right, then Dr. Bruno, I think you wanted to...
Well, you know, it's a fascinating discussion. I think we could stay here all night. But the one thing you were talking about, or Tim, I think you mentioned, that autonomy being such a central concept, but is it such a central, I mean, is that evolving? Is autonomy not going to be the way we have defined it? Is it more of a collective consciousness? Are we moving to a point that we aren't this individualistic? Is that a different value that's coming on a fore? Is there the merging of inanimate with animate, you know, in a sense, or inorganic with organic, cyborgs. I mean, and I am worried about the social justice issues because I think, but that's not, I think that may stretch the concept of autonomy, even social justice. Possibly.
You know, we want ethical concepts that protect what's important. Right? And I think, as Professor Prime mentioned, you know, the dignity of the human person is important. And I think autonomy is the concept that best protects human dignity. Right? So if we get rid of autonomy, like what are we dealing with? Right.
I know, but I don't know.
But we might be getting rid of it. So I think...
I don't know if we're time bound with, I'm just throwing it out. When you listen to this, I think we're really at an incredible crossroads. You know, we often talk about artificial intelligence getting to a point far superior to human intelligence. And so I think so many questions are coming, and is the future both exciting and scary at the same time, and is autonomy going to be eroded?
Yeah, so I also think our ethical concepts haven't caught up, right? So our ethical concepts like autonomy, they're from the 17th to 18th century, right? Yeah. So maybe we need to invent new ethical concepts to discuss these issues.
Now, before we leave, I want to thank everyone. Michael, all right, one thing.
I don't know if this may have been thought, but is this we don't have any other examples of life since we haven't been contacted by anyone. But perhaps this is our natural evolution.
Could be, could be.
Yeah.
Let's end with that comment.
Well, I just want to know that I thought about this last night, that we say the future is now, but what people don't realize is that when you do make a change in a protein, even though you've improved it, how is it going to affect the interaction with others? Because that's how cancer arises. You get mutations in one, maybe two genes. It changes the protein from those genes. And then their interactions with others, other normal ones, sets off the cancer process. All right? So even though we're changing our protein, this one gene to make this one change in the protein, and we say, oh, it's improved, how is it going to affect the rest of us? So actually, this technology, though we can do it now, the consequences are still kind of far away. All right, still kind of far away, because we don't know half of what's going on inside here. We're truly children playing with fire. That's what we are, okay? So I want to end on that. Thank you everybody for showing up.